My name is Natasha, and I will be talking about this is my homework. I will be talking about name one benefit and one challenge to using genetic method to identify microbes. So I went on to explain that microbes are tiny living things that are found all around us and are too small to be seen with the naked eye. They live in water, soil, and in the air. The human body is home to millions of these microbes too, also called microorganisms. I got this information from the National Center for Biotechnology Information. So some microbes make us sick. Others are important for our health, the most common type of bacteria, virus, and fungi. There are also microbes called protozoa. They are tiny little things that are responsible for diseases such as toxoplasmosis and malaria. So some microbes, including bacteria, fungi, parasites, and viruses, cause infections in humans. They are known as infections, agents, or pathogens. Traditional testing techniques for detection of pathogens includes, for example, growing microbes in cultures, followed by identification of the microbe or testing blood samples for antibodies that people develop in response to an infection. So I want to give a little example on how to collect, to use the proper, successful identification of microbe and how to collect it. So first of all, you have to use the proper equipment when you're trying to obtain the specimen. So these here are a pair of gloves. I've also included a cotton swab, and this is we're going to be checking a patient for yeast. This is once it's collected by the provider with this cotton swab, and she will have on her gloves. Once it's collected by her, then she would put it in a specimen. After the specimen, after the specimen, then we put it in a biohazard bag and we send it to the lab so that they can go ahead and run it properly. Because the microbe contains genetic material, also called nucleic acids, the DNA and RNA that is different from the techniques can also be used to detect microbes. So basically, the genetic testing may be more sensitive and specific than traditional methods of testing. And like I said, the bacteria that is usually sent off so they can check for yeast, they can check for chlamydia, things like that. The virus, that one, when they collect things to have the virus checked, that's sent off and that can be checked for the flu or COVID-19. Usually the treatment for these are antimicrobial and medications that are used to stop the growth or to kill the microbes, or antivirals and antibiotics are examples of that. The benefit to all of this is so that you can the proper treatment is given to the patient depending on the growth of the microorganism so that it is treated. This information has come from the United States National Library of Medicine, National Institute of Health. Thank you.